This Week in Virology, the podcast about viruses, the kind that make you sick. Hello, Zurich. They're much more restrained than American audience. From Microbe TV, this is TWiV, This Week in Virology, episode 523, recorded on December 6th, 2018. I'm Vincent and you are listening to the podcast all about viruses. Today we're recording in Zurich, Switzerland. I'm on the campus of University of Zürich. And I have gathered three virology professors here to talk about their careers and their work and their research on viruses. And we have a little audience. Thank you for coming to witness science communication in action. My guests today, here right close to me, Professor Silke Stertz. Welcome to TWiV.
Thank you.
Did I do a good job with that?
Yes, you did a very good job.
I got some coaching, you know. Next to Silke, yes, next to you is Professor Cornel Fraeful. Welcome to TWiV.
Thank you very much.
Is that good? Fraeful?
Perfect.
Okay. And finally down there at the end, Professor Urs Greber. Welcome to TWiV.
Thank you very much, Vincent, for having us.
My pleasure. Thank you for doing this. I know it's something a little different that you're not used to, but it's really easy for you because all you need to do is talk about yourselves. And we all like to do that, right? So there's no problem. I'm the guest of the LSSS, Life Sciences Seminar Series. Is that right? Did I get that right? Who invited me here, and I gave a talk yesterday, and we decided to record an episode while I'm here. Might as well coming all this way. It's our intention to let the world know about the kinds of virology that's happening everywhere. So that's why we do this. So we're going to chat with these individuals today about this. Now, a lot of people don't like when we talk about the weather on TWiV. I get emails almost every day. In fact, just this morning, I read an email from someone who said, please skip the weather. However, it's so bad today in Zurich that I have to mention it. In fact, it's been cloudy for my whole trip. Today, that's the weather here. Zurich, it's 8 degrees Celsius. It's cloudy and it's raining on and off. So, does the sun ever come out here in Zurich? Yes, it does. Okay, there you go. All right, I want to start by finding out from each one of you where you're from, where you went to school, college, graduate school, postdoc, etc. And let's start with you, Silke. Where are you from originally?
I'm from a very small town in the southwest of Germany, and there I went to school, and I then went to the closest university, which is Freiburg in Germany. I studied biology there, and then ended up in virology and really liked it.
And then you decided to get a PhD.
Yes, I did the PhD also in Freiburg with Otto Haller and Georg Kochs, working on the innate immune response to viruses, mainly on MX proteins and their inhibition of influenza viruses. And there I realized that the influenza viruses were really what I'm most interested in and what I really want to continue working on. And then I looked for postdoc positions. And ended up in Peter Palesi's lab in New York. And I think that was a very good
What year was
decision. That? I went to New York in 2007.
Okay, so I was Peter's first student.
I know.
In 1975 I started.
Okay.
It was before you were born. Yeah, I posted a picture of myself in his lab in 1977 and someone wrote, oh my God, it was before I was born. Like, yeah, that happens. What can you do? And then after that postdoc, did you come directly here to Zurich?
Yes, in 2011 I started setting up my own group here in Zurich.
So how far back can you remember liking science? Do you know why?
In school, I just enjoyed the science, like chemistry, biology, also some math, and I found it really easy. I didn't have to study very hard. I just liked it and found it very interesting, and I thought that was a good choice to go on. I didn't really want to become a doctor, that I considered for a while, but no, it was science, and then I studied biology and really liked it.
Great. Well, did you enjoy your years in New York City?
Oh, yes.
It was fun,
Yeah.
right? Yeah,
Yeah, that was a very good time. Yes.
Cornel, where are you from?
I'm I grew up in a small town, eastern Switzerland, close to St. Gallen, where I also went to school, to high school, studying mainly Latin and philosophy.
Can you say something in Latin?
Ave, Vincent. But then after that I decided I want to do something useful, went to the Federal Institute of Technology, the ETH Zürich, where I studied biology. I also did a PhD at the ETH on bovine herpesvirus 1. Then later I went to a first postdoc in Boston, the Children's Hospital with Alfred Geller, and the second postdoc also in Boston at the Mass General Hospital with Sandra Brakefield, where I got into this gene therapy, virus gene therapy field. And then later I came back to Zurich, where I started my own group at the Institute of Veterinary Virology, starting my own group on studying the interaction between co-infecting viruses, specifically adeno-associated viruses and herpes simplex viruses.
Okay. And then you started your lab here.
Yes.
And you've been here ever since.
Right.
So I went to Cornell University, you know.
Yeah, I think they misspelled the name.
Yes. You have one L in your name, right? Yeah, I got it. So far we have two panelists who spent some time in the US. Let's see if that holds for all three. So Urs, where are you from?
I grew up in a city 20 kilometers north of Zurich, Winterthur, and that's also where I went to high school. And then I started to study at the University in Zurich, at the ETH in this time. Experimental biology was called at the time. And I was doing a master thesis on cloning and isolating a gene encoding creatine kinase that is involved in muscle development. And then I was interested in genes and how they work, and I think I was influenced quite a bit by Charles Weismann at the time, who was really incredibly fond of finding out of how genes work, and it's one gene, one protein hypothesis really captured me. And I then went into a PhD thesis at the ETH, at the Institute of Biotechnology. And it was a little bit of a different subject there. It was about genetic engineering of yeast cells in order to make them secrete certain proteins of interest. But it turned out that this project was quite complex and complicated because yeast cells have a different way to glycosylate their proteins than human cells. So if you want to use therapeutically proteins made in yeast, you have an antigenic problem. And the project of my PhD thesis was to deglycosylate and try to reglycosylate these proteins made in yeast. That was a tough job, which was not hugely successful. And but I got great advice from a fantastic cell biologist who I was fortunate to train with in the last year of my PhD, not at ETH, but at the Swiss Cancer Institute in Lausanne. His name is Howard Ritzman. He's a yeast geneticist, and he pointed me to the field of nuclear transport. And then I went for a postdoc to investigate this topic in the lab of Larry Gerais at Scripps Research Institute in La Jolla, San Diego. And there became fascinated with the problem of how proteins communicate between the cytoplasm and the nucleus and how they traffic. And Larry was then kind enough to advise me further into my second postdoc, which was then done in the lab of Ari Helenius at Yale. And the motivation for doing that switch from the West Coast to the East Coast for me was that I wanted to have kind of a niche to explore how nuclear cytoplasmic transport works. And Ari's background on viruses, and that's why I'm in viruses now, was exactly on this question, namely the cell biology of virus infections. And that kind of combined my interests and also my wish to work in a kind of a small protected area at the time and try to explore this problem a bit. And so we started with the big question, how can an incoming virion, in this case the adenovirus with its DNA, get its DNA into the nucleus? That was a big, big question. People knew that this was possible to do because earlier data indicated that the incoming viral DNA, which was labeled with nucleotides, was indeed over the nucleus. But how it got there was unknown. And that was what fascinated me so much about viruses.
And then after that, you came back to Zurich, and you've been here ever since, right?
Yes, then I was fortunate to get an assistant professor fellowship. At the time it was called START. It's a Swiss award for in academia and in teaching. It was a non-tenured track position that I intended to start in Charles Weissmann's lab, but at the same time I was also applying to a tenured track position at the Institute of Zoology, and there they were looking for a cell biologist, and I was fortunate and lucky enough to get that position as well, and so I decided to set up my group at the Institute of Zoology and pushing that. That was a few buildings next to molecular biology and Charles Weissmann, but it was not in his lab.
Do you remember where your interest in science came from?
Yes. Yeah, so maybe that goes back all the way, maybe goes back to my father. So my father was always very fond of nature, and he was an engineer, in fact. And he was teaching us to look at things in numbers also. So when we were hiking out there and saw these big rocks in the mountains, for example, he was asking us, do you know how heavy this stuff is? And or do you know why trees are not growing indefinitely into the sky? And those sort of things. And that was only, that's a good question. So how should I find out? And he tried to explain me that. He didn't know all these things, of course, in all the details, but he was, waking up a kind of a curiosity inside me, and that kind of kept me going in a way. So trying to find out how things work the way one can observe them.
So here we have 3 individuals who spent some time in the U.S. So how about the audience? Who spent time studying in the U.S.? Okay. Who plans to spend time studying the U.S.? Maybe a few as well. All right. What do you like better, the West Coast or the East Coast?
It was fantastic to be on the West Coast. At the time, I had the feeling of being on vacation. It was so relaxed, and you didn't get the idea of having to work in the lab. But you did, of course. And things just happened somehow.
Yes. Now, California is nice.
And this was just incredible.
Cornel, do you remember where your interest in science comes from?
Well, also my father, he made us interested in nature. We were always in nature. By the time I was in kindergarten, I was carrying snails in my pockets and worms, bringing them home, dead squirrels, birds, everything. I was really very much into biology.
I'm always interested to see how people started with an interest in science. And both of you said your father, which is interesting. And my father also, he was a doctor, and it got me interested in medicine anyway, but like you, I didn't want to be a doctor. Yeah. Absolutely not. So it was hard to say no to my father, who wanted me to be a doctor, of course. So Cornel, let's talk a little bit about your research. You sent me a paper on adeno-associated virus 2 dependence on herpes simplex virus type 1. So let's talk about that. First, tell us a little bit about AAV2 and the dependence before we get into the paper.
Well, AAV2 is a very small single-stranded DNA viruses and depends, unusual for a virus, on a second virus to complete a full replication cycle in the infected cell. So I was fascinated by the situation where 2 viruses are in the same cell, manipulate each other, one of the viruses even needing the other one, and in fact able to completely manipulate the large one, the herpes simplex virus, which has a very large genome, many different genes, but nevertheless the small AV with only 2 groups of genes can completely control the replication of the large one. So we were interested in studying on which level the two interact. We looked at mainly at replication in the cell nucleus, like asking questions like, herpes simplex virus is a very large virus with many genes encoding for enzymes for DNA synthesis, which allows him to be very autonomous from the cell, to replicate in all phases of the cell cycle, versus the very small one, which is completely non-autonomous. So we ask whether HSV1 as a helper virus can extend its ability of replicating in all phases of the cell cycle to AAV2. So we started these experiments looking at AV replication compartments in live cells, AV gene expression in live cells relative to cell cycle phase.
Now, in nature, what would be the situation? You would have, we're all infected with herpes simplex 1, so then AAV2 would then infect those cells? Is that, because they're not going to infect at the same time probably, right?
I get this question very often. The question is indeed, is HSV1 indeed a helper virus in infected
people? Yeah.
Or not just a helper virus in cell culture? Because the cells they infect are very different. Although there may be one tissue that would be interesting for both viruses, these are like places where latency of HSV1 occurs, ganglia of sensory neurons, and I think that would be the tissue to look at co-infection with the two viruses.
Has that been done?
We have looked just recently at 3 tissues from humans and have not found AAV2. We have found HSV but not AAV2.
Have people done serological surveys for AAV2?
Yeah, I think most of us are AAV positive. Yeah.
Okay. So what HSV1 functions are needed by AAV2?
It's basically functions that are relevant for DNA replication. It's ICP8. ICP8 is a single-stranded DNA binding protein that is essential for HSV replication, but also for AAV replication. And the other component is the HSV1 helicase primase complex.
Okay. So AAV, as you said, only encodes 2 proteins, a capsid.
2 classes of genes. Yeah, it's the rep proteins. There are 4 isomers by alternative splicing and 2 different promoters, and 3 different cap proteins.
Okay, and the rep is involved in a couple of functions, including DNA replication, right?
Replication, integration of the AAV2 genome into the human chromosome 19.
Right.
Genome packaging into capsids, so it has a lot of different functions.
By the way, chromosome 19 is one of my favorites because the poliovirus receptor is on chromosome 19.
Right.
We've known that for many years. So whenever I hear 19, I perk up. So if you take just AAV2 and infect cells, where does infection stop? At DNA replication?
It stops likely after second strand synthesis and small levels of gene expression.
I see.
The promoter that is used for rep gene expression is very inactive in absence of a helper virus. But you do get second strand synthesis and low level gene expression.
So when the genome is single-stranded, it goes into the nucleus and second strand synthesis then occurs. What is that done by the cell polymerases?
Actually, we don't know. We have looked at that, which polymerase is responsible for this, but we do not know at the moment.
Yeah, my students always ask me what that first one, is that a repair thing or is that a replication? You probably look at it as repair, right? DNA repair.
Could be either.
Right.
Yeah.
So in this paper you sent me, which is a JV paper, cell cycle dependent expression of AAV2 rep in co-infections with HSV1 gives rise to a mosaic of cells. You look at this paradox, which is how do 2 viruses co-infect and not inhibit each other. So tell us how you did that.
So we use these HeLa Fuji cells. They express two different fluorescent colors linked to cellular proteins that are expressed in specific cell cycle phases and in other phases are degraded by proteasomal degradation. Specifically, we used geminine fused to a green fluorescent protein and CDT1 fused to a red fluorescent protein. So the cells appear red in G1 and in S phase, in G2 phase and M phase they appear green. And there's a very short period between S and G1 where they are at early S phase where they have both colors and are orange.
So if you look at a plate of cells growing, you can see them cycle through these colors. You must have made a movie of them.
Yeah, we have many nice movies where you see them go through these colors.
Are they on YouTube?
Pardon?
Are they on YouTube? Your movies?
No, but they are, actually you can access it from the Journal of Virology.
That would be great. We'll put a link to it.
Great,
People would love
good.
to see this.
Great.
Fuji, is that who made them?
No, we didn't. No, it was made by Sakaue in 2008. Was a Cell paper.
Okay, I think these are amazing, and some of you use these cells. I think Urs, you use them in your laboratory, right? So, what did you use them in the context of co-infection?
So anyway, we had three colors already occupied: orange, green, and red. We had to make another color to visualize either AV replication or AV gene expression. So for AV replication, we fused a blue fluorescent protein to the rep protein, which is a DNA binding protein. So by binding to the replicating AV DNA, this allowed us to visualize the growing AV replication compartments. And when you use just a blue fluorescent protein encoded by a recombinant AV vector, this is for visualizing AV gene expression. So we used the Fuji cells, infected them with the recombinant AV, either expressing the fusion rep blue or only blue, and infected them or superinfected them with herpes simplex virus as the helper virus. And what we found was that both AV replication but also AV gene expression occurs predominantly in G2 phase. And we also found that when AV2 co-infected cells, HS1 replication becomes restricted to G1 cells. It was not able to replicate in G2.
Normally it would replicate all throughout the cycle, right?
Right. So we initially thought that maybe second strand synthesis is a limiting step that may not occur in G1 but in G2 cells, so like a licensing factor in G2 cells. So we used something called a self-complementary AV2 vector, which is a AV2 vector that has a genome that can form immediately a double-stranded DNA by self-annealing. So we skipped the step of second strand synthesis, but also this vector was predominantly expressing in G2 cells.
So the solution to the two viruses are that they are replicating in different parts of the cell cycle.
Exactly.
Now, do you know, so what causes this? Do you know?
We would like to know, so we look at different things like AV circularization, AV trafficking, uncoating. At the moment we do not know which is the step that is causing this cell cycle dependence.
So, and also, the reason you looked at this initially is because AAV would inhibit HSV synthesis, right? Is that caused by rep?
Exactly.
And how does that work?
So we have one study where we show that the AV rep protein can directly bind to rep consensus binding sites on the HSV1 genome. And we showed that AV rep can basically inhibit the replication of any DNA as long as it can bind to it. So we placed consensus rep binding sites on plasmids, and we showed that replication is inhibited by rep. based on these consensus sequences.
Right. So it just happens to be binding to sequences that are present in the herpes simplex genome.
Yeah, there are approximately 9 such consensus binding sites.
The other thing you say in this paper is that rep is only made in SG2, right? And that's why the virus replicates during that time. Do you know what restricts the synthesis of the protein to that part of the cell cycle?
Well, at the moment, we look into uncoating, and perhaps, we need uncoating before we get second strand synthesis and gene expression. But we have not come very far with that yet.
So back up a little. When AAV2 infects the cell, does it stimulate the cell cycle? Does it stimulate cell division? Because many viruses do, right?
What our observation is that it does not stimulate cell cycling or progression of the cell cycle, but it induces an arrest in G2. As at least with the single-stranded recombinant virus as well as with wild-type virus. However, I mentioned earlier the self-complementary double-stranded virus. This one also, although it requires G2 cells for gene expression, it does not arrest the cell at the G2 phase. It goes over cell division. And we think that this may be related to the single or double-stranded nature of the DNA, which may induce DNA damage responses that cause cell cycle arrest.
So there are some there's some parvoviruses that are not helper dependent. How do they differ so that they don't need a helper from AAV2 set?
Like B19 is a human parvovirus that does not depend on a helper virus. People have at one point replaced the AV2 P5 promoter or placed the P5 promoter into B19 and made it helper dependent.
Promoter.
It's probably related to some specific sequences.
Right. So AAV vectors are widely used in gene therapy. Does this result have any implications for that?
Well, I think if you would think of how to produce AAV vectors, of course G1 cells are completely useless to make AAV vectors. So I think you could probably manipulate or maybe synchronize cells to produce more or high-titered vector stocks. Yeah. The other thing is that most targets of gene therapy are non-dividing cells.
Right.
So often they are in G1. So obviously, and we know that AAV vectors are not very high, don't have a very high transduction efficiency. So knowing now that it is related to G1, one could have ideas how to modify the vector so that it also transduces G1 cells more efficiently.
Do you ever think about how, so let's assume that this dependence occurs in nature, even though you said you're not sure, which by the way, it doesn't matter to me because I think it's interesting to find out why one virus would be dependent on another. But let's say it occurs in nature. How would this have evolved? Do you ever think about that?
There are reports or there are papers where they looked at co-evolution of the helper viruses, specifically herpes simplex virus and AAV, and people really think it's a result of the co-evolution of the two viruses.
So at one point it was AAV may have been helper independent, and then it evolved to lose that function?
Perhaps. There is one very interesting herpes virus, that's human herpes virus 6.
Yeah.
It has a rep homolog, and to my knowledge it's the only herpes virus that has ever been shown to integrate into a host cell genome. So there are links between the two viruses.
Yeah, I mean, I always like to think about how things evolved to the state where they are now. Sometimes it's very difficult to imagine, right? But that's cool. Okay.
And in fact, if I can add this, this site on chromosome 19 where AAV integrates, contains a rep binding site and a terminal resolution site. That's the site where the rep protein introduces a nick. So it could very well be that AV was created from this spot.
Right.
Maybe with the help of a helper virus.
Interesting. All right. We'll come back later. I have some other questions for you. But Silke, let's talk a little bit about your work. Looking over your publications, seems like you like to use screens to identify things that are important for influenza virus.
Yes.
So let's talk about some of those. I think that would be interesting for... You did your first one with Peter, right?
Yes. I had a fellowship from Germany for my postdoc in Peter's lab, and I had planned out my project. I wanted to study RNP transport to the plasma membrane. Was quite proud of that project. And the first week I started there, and Peter said, I've got a different project for you. But I spent so much time planning that, and he was like, no, this is really exciting. You're going to do that screen. And so in the beginning, I wasn't sure, but you don't really say no to Peter. So I started working on that screen, and as usual, he was right. It was very exciting. So what he had been setting up with collaborators was to do a genome-wide RNAi screen, where you downregulate the expression of almost every cellular gene, and then see what impact it has on influenza. And there was this idea, and there was a collaboration partner, Sumit Chanda in San Diego, who had experience on screening for HIV host factors. And so we worked with them and established a screening protocol, and then ran the screen and identified host factors that influenza virus needs for efficient replication. And the idea was that, first of all, you can understand more of the cell biology and molecular biology of the virus and reveal these interactions with the host cell. But one idea was also that maybe this could be a strategy to develop novel antivirals, because the virus has very few proteins that it encodes, with even fewer enzymatic functions that you can easily inhibit. And so we thought maybe this could be explored if this is a strategy to expand the range of potential drug targets. That was the idea.
So did you bring back from his lab some of these hits and start to work on them in your lab?
Yes, because one issue with the screens, there have been many screens for many viruses, and the initial result that there are lists of factors is interesting, but it doesn't really tell you much. The follow-up is where you really learn and where you can then identify promising drug targets. And so my plan was to come here and set up my own lab and then characterize some of these factors to really understand how the virus uses them and if there are enzymatic functions, for example, that could be inhibited.
So one of the papers, I think, and this is a result of that screen, it was identification of polo-like kinases as potential novel drug targets. Is that a result of that screen, or is that something you did here?
So initially from the screen, we mostly worked on some proteases, peptidase, and Cathepsin W. But when we also had a project in the lab, because there were these different screens done by different labs, and sure, there are different experimental conditions, so you would expect some differences, but overall there was little overlap between the screens. It was really surprisingly little overlap, and we thought that's really an issue that we need to understand better. Also, to give it credibility, or to really show the field that these data can be useful. And there we worked with several of the groups who had these screens and tried to get the primary data sets, because what we thought and what turned out to be right is that a lot of the differences in these hit lists come from the bioinformatic analysis of the screens. Everyone uses a different method, everyone uses different cutoffs, and we could really show if you use the primary data and analyze them all in the same way, you get a much bigger overlap. And then when looking at those data, we saw that these polo-like kinases kept showing up on the different hit lists. So that was a study that came out of more the meta-analysis of the
So what is a polo-like kinase?
They are actually very important in cell cycle regulation. But there are different ones, and they have been involved in different functions. And some of the members are quite essential and might not be really a drug target for the side effects, but they kept coming up, and so we really wanted to explore if they are required for the virus, and we found that they do matter, and there are inhibitors. That is always a useful tool to study. But at the moment we're more at the stage where we're exploring different factors that came out of the screens and try to understand more their molecular or cell biology function, and then from that we have to see which one looks the most promising.
So in these kinases, these are serine threonine kinases,
yes,
right?
Yes.
Do you know what they phosphorylate? They have clearly cell targets, right? Is that known?
There are long lists
okay.
of targets, many of them related to cell cycle regulation.
Are there viral targets as well?
We did not see any changes, but that I think doesn't mean that there aren't any. There are certain limitations to the sensitivity of these assays.
Do you think the inhibition, so if you knock down these polo-like kinases, you get less influenza replication,
yes,
right? Do you know where in the replication cycle that affected?
Mm-hmm. That was an early block, so we mostly focus on the factors that have a role early in the replication, and they also came up in these when we do sub-screens for the entry factors. They always kept coming up, and we could confirm that with more specific assays that it's really early, but we don't know exactly which step of the entry. We have other host proteins or other factors where we've gone a bit further and can map more precisely where the virus requires.
So when you say early, you're talking about entry, right?
Yes.
So in a polo knockdown, does the RNP release from the endosome, or is it blocked?
It does release, but then we do end up with less RNPs in the nucleus.
Got it.
But where exactly in between that we couldn't, or we haven't further explored for those.
Do you have an idea of what it might be?
No. No.
But if it's a cell cycle effect of these kinases, it could have to do with nuclear membrane breakdown. Permeability,
yes.
Maybe.
Yes.
But the RNPs can get through the pores, though, right? So.
Yeah.
But it could be regulated.
Looked into that.
So another hit was this Cathepsin W.
Yeah. Mm-hmm.
So what is a cathepsin? What does it do in the cell?
And we were quite intrigued to find cathepsin quite high up on the hit list, because cathepsins have been implicated in entry of many different viruses. And usually they cleave the viral glycoproteins. Right. And so we thought, this is interesting that we find this slightly weird, I should say, cathepsin where not much is known about. It's not the usual ones that you find, S or B usually for the other viruses. But we got interested in that, and that is something where we've mapped the requirement for the virus a bit better, and where we still work on. And what we could show is that cathepsin localizes to the endosome, or late endosome in particular, and that the virus really gets trapped in the late endosome. So virus binding, internalization, endocytic traveling, that all happens normal if you downregulate Cathepsin W, but then the virus is really trapped in the late endosome and fusion is inhibited. And that is where the virus needs it. And we could also show that this requires the catalytic activity. So if we do knock down and put back the wild type, we can rescue the phenotype or the block, but we can't rescue with a catalytically inactive mutant. Okay. So we think this could be a potential drug target that we want to explore, and that's something we're working on.
And that result tells you that something is being cleaved.
Yes.
But it's not the glycoprotein, right?
We did not see any glycoprotein cleavage in the assays we have done so far. So at the moment we think it's probably a cellular protein, but one hypothesis we had is that it could also be for the fusion process. You might need quite some space, and the envelope of influenza is packed really densely. Yeah. So could it be that locally at the fusion site the protease removes some of the glycoproteins to make space for fusion? But we haven't seen that in Western blots, but it could be that only 2-3% of the total amount of glycoprotein is cleaved, and you might not see that. So this we probably can't rule out either. And we're now working here with the proteomics facility using a proteomics approach to look for cellular targets.
So you're looking for cell proteins that are not cleaved that could be involved in this.
Yes.
But maybe there's, as you say, some viral proteins that are cleaved as well, right? That could be.
Yes, we're doing the proteomics assay with infected cells, so the hope would be to pick up either of those two options, but we don't know at the moment.
The other thing I wanted to ask you about was this phosphoproteomic-based kinase profiling, which your paper says identifies GRK2 as a drug target. GRK2 is G protein coupled receptor kinase 2. So what is quantitative phosphoproteomics?
So what we did there is we infected cells for a very short time, 5 or 15 minutes, and then from the cell lysate we isolated all the phosphoproteins, the phosphopeptides, and those you can measure with mass spectrometry, and you can quantify the levels. And then we looked for phosphopeptides that were either more abundant or less abundant in infected cells compared to mock-treated cells, and to see what kind of signaling cascades get activated upon virus infection. And there was evidence before that just the virus binding by itself already triggers all kinds of signaling, EGFR, MAP kinase signaling, and this is what we then also saw, plus novel factors that seem to get phosphorylated or dephosphorylated in response to infection.
So if you look at these peptides, can you tell what kinase is responsible for the phosphorylation?
The bioinformaticians can, yes. If they look at the whole list of peptides, they can then deduce some motifs that are overrepresented, and that can then be linked to kinases that prefer those motifs.
So is that how you identified GRK2?
Yes, yes.
And so then you knocked it down and showed it was needed for replication?
Yes, exactly. And then we could map the requirement to the uncoating. So up to fusion, the entry process occurs normally at the same efficiency as in control cells, but then when you have GRK knockdown, the uncoating, so the release of the RNPs is inhibited. And again, we don't know the target yet. It has been described that GRK2 phosphorylates HDAC6, and there was a very nice paper from Ari Helenius not long ago that HDAC6 is really important for uncoating of influenza virus. So we thought, oh wow, that's it. But it turned out that these two things are not really linked, that the GRK2 has no impact on the HDAC activity, at least in the assays that we have done. So we still don't know how the kinase mediates this.
So do you know all the targets of GRK2 in this cell?
No. So there are some that are published, and we have some peptides that have the GRK2 motif, but from the top ones that we have tested, we didn't see an impact on uncoating, so this is still ongoing that we're looking. Yeah. And the problem is with this phosphoproteomics, you have only a snapshot. It's like one time point where you look at what peptides you have, and this could have 3, 4, 5 downstream events that you wouldn't then see. So this is still ongoing.
Of course, these are in cultured cells, cell lines with certain strains of influenza virus. So you could say, well, if you looked at respiratory tract tissues, you might find something different. Do you plan to look at any of these in like bona fide?
That would be great,
tissue?
But at the moment the sensitivity of the method is not yet there to do that with like small amounts of cells from the differentiated cultures. So the idea is more, what we're doing now is we're repeating the proteomics with more primary isolates from humans and also avian isolates. To see how that compares to the lab strain, and when we then have the interesting candidates to then look on a smaller level or smaller scale in the differentiated cultures.
So do you have an idea whether the protein target of GRK2 is within the endosome or outside the endosome?
I would guess outside. But I don't really have data for either of those two, I have to say.
Now, in each of these papers, you always say this could be a drug target, right? So, of course, the basic science is fascinating on its own. So are you interested in pursuing these as drug targets?
Yes,
or?
I think so. Not every single host factor that we look at will turn out as drug target, but I think at this stage it's important to characterize some of them and really understand what they are doing to then go on and maybe like develop compounds first of all. And I think this Cathepsin W is a good candidate. There is a knockout mouse that is perfectly viable. I know men are not mice, but it still indicates that maybe this is not too essential and that one would have a chance to develop inhibitors against that. So that will be a next step when we understand what this protease is doing, that we run a compound screen for Cathepsin W inhibitors that we could then test.
All right.
Cell culture.
So at some point you have to go into an animal model to get the data and then proceed from there. Well, certainly we do need more antivirals for influenza.
Yes.
But I think the mechanistic information is really fascinating too. So, you know, it's always, in the US, we always have to add these statements to grant applications. You know, this could result in a drug target, which I find unfortunate because I think the science itself is fascinating and it always leads to something interesting. And you know, to do this targeted statements, it's not your fault, it's just the fault of the system. I find it's unfortunate. But all right, we'll come back to you. Let's talk to Urs a little bit about your work. Now, your work, I remember from Ari Helenius's years ago, and in fact we put it in the textbook very early in the first edition. I wrote the chapter on virus entry, and I remember reading your papers on adenovirus entry because you had done those early experiments, as you said, to understand how a DNA virus gets its genome into the nucleus. So maybe you could start by summarizing adenovirus entry. What do we know from the cell surface to the nucleus so far?
Yes, so I think the concept is that the virus uses different host cues to start its disassembly program. And this is a very important job for the virus to do, if you will, because it has a very well-protected genome in a capsid that is sturdy and resistant, and it requires different cues. So we look at this as a gain of function for the virus, for the virion, as it travels along the entry pathway from endosomes to the cytoplasmic compartment on microtubules towards the nucleus. So it progressively loses components, and the human adenoviruses they are built in a modular way such that it has major capsid proteins and also smaller proteins, so-called cementing proteins. And Roger Burnett at the time has coined this name, and I think it's a very useful name because they reflect both the nature of these proteins and also their function in a way. And the virus gets rid of them in a stepwise manner. And that was essentially the concept that we put up in 1993 when we analyzed the adenovirus entry from the cell biology point of view. Now we know and start to know more and more of these entry helpers from the cell. So they are like, you know, in the Trojan horse, Troja, right? So they're the traders in Troja, right? That instruct the horse on when to open up and release the cargo. And those sort of things we start to explore and know more and more about. So we like to look at this problem in a single cell manner and also in a single virion manner. So where we track the virions with fluorophore dyes, each of the particle is visible in the fluorescence microscope, and we can use a time-resolved analysis of that, and we can see where it is at which time point and what kind of protein it is associated with it. And that kind of concept is very powerful because it allows you, for example, to clarify certain myths in the field. So there was the notion that the virus was pH dependent because of some interference experiments and some bulk biochemistry experiments. But when we started to look at this by the single virion assay, we noticed that the virions get out of endosomes even in the absence of low pH. So there's something else happening there that we need to explain why certain lysosomotropic agents inhibit the virus penetration. So this is an unknown. We suspect that these are other ions in endosomes that work on the virion, but we don't know yet what they are. Things like that help us to molecularly describe these different steps that the virion is taking as it engages with the cell machinery.
How does it get out of the endosome before the pH drops?
Yes, so the model there is that it uses mechanical cues at the plasma membrane to open up its capsid. And so, like almost any other non-enveloped virion, I think correct me if I'm wrong, the membrane lytic protein is usually hidden away. It's either in the capsid wall or even inside the virion. So the same is true with the adenovirus. The membrane lytic protein is inside the virion. There's 340 copies of this thing, and it's hidden. So the virus has to mobilize this somehow towards the outside. And the way he does that is he is opening up the capsid by pulling on its fibers and holding back the virion with another receptor. And this mechanical force, we think, opens up, we have evidence, direct evidence that this happens, and opens up the virion to release 6. And now 6 binds to the plasma membrane. It doesn't bind that well because it prefers a certain kind of lipid to bind, which is ceramide. And normally the plasma membrane doesn't contain a lot of ceramide because that's a cell signaling a cell death lipid that is also associated with lipid rafts. But the virus or the virion has found a way to increase the ceramide levels at the plasma membrane, namely by triggering lysosomal secretion. And that releases an enzyme, among other things, that cleaves sphingomyelin. It's a lipid precursor essentially of ceramide, and now increases the ceramide levels on the plasma membrane. And now as protein 6, more and more protein 6 gets released, it binds more and more avidly, better, and together with the endocytosing virion, it is now in a endosome that is prone to open up its membrane. So we believe there is a biophysical disturbance of the lipid flow that is impinged on by the amphipathic helix of protein 6 that disturbs the lipid integrity and opens up the endosome.
So we have to change the textbook, because in the textbook we say it's a low pH dependent exposure of these lytic proteins in the virus particle, but you don't think that's the case anymore? Yeah.
Yes, so I think this is part of the old school of looking at things. And it came from the experiment where people classically looked at which of the virion proteins would segregate into the lipid phase at low pH, for example. And there they found that certain proteins do that. And based on that, they came up with the notion, yes, that virion must be pH sensitive somehow because it kind of, puts these proteins in there. But when you look at the real data, there's little evidence actually that this happens in the cell.
Okay. So it's good because we're just writing the next edition. So we'll fix that. So then the virion is partially disassembled, gets out of the endosome. What happens next? Yes,
the virion now is in a position to pick up microtubule motor proteins. And Richard Vallée and others have shown conclusively that there's a dynein directly binding, not through dynactin complex, but through another parts of that motor protein, directly binding to the virion. And now this is now helping the virion to travel on microtubules towards the minus ends. And since the minus ends in these cultured models are close to the nucleus, it already is an advantage for the virion now to be closer to where it wants to end up and deliver its genome.
So it's close, but it's not exactly there, right?
Exactly.
So how does the last step happen from when it falls off the microtubule? How does it it needs to find a nuclear pore, right?
Yes. So we have been puzzled by this problem quite a bit, actually. And until some time ago, a student in the lab found that when we inhibited nuclear export with a drug called leptomycin, then the virions kept traveling on microtubules. They were not able to detach from them, and they were not able to reach the nuclear pores. They were perfectly reaching the microtubule organizing center, but they were not able to detach. Now, follow-up study on that one then has shown that the virion requires a cue from the nucleus that is only available in a spatially controlled manner near the nucleus, within maybe 1 to 2 micrometers. And that cue, we don't know what it is exactly, but it coincides with the notion that the virion keeps traveling on microtubules back and forth if this cue is missing. So we believe that this detaches the virion from the microtubules and allows it, by an unknown mode of action or mode of motion, to reach the nuclear pores. And there the nuclear pores work as a sink, right? They have an affinity for the virion, so you enrich the virions there.
Okay. By the way, could you push the microphone forward a few inches? We're getting a little feedback. Okay. So now that the particle's docked on the pore, the DNA has to get in the nucleus. How does that happen?
Yes, this is a complex task for the virion because it is still an entity that is enclosing its DNA genome, and it itself is too big to get through the pore. So it's got to separate the DNA genome from the rest of the capsid. And inside the virion, there are proteins that condense the DNAs, we think, or organize it somehow. Maybe I shouldn't say condense, but organize it somehow, and others that link the DNA to the capsid wall. And those links need to be released somehow. And a very exciting project in the lab. It's not published. It came from a screen, in fact, an RNAi screen, in fact, one of many hits that turned out to be a very interesting hit. A protein, a ubiquitin ligase that is working on the virion to make this happen. And then there's a second step happening, and that's a published mechanism, where a kinesin motor protein is binding to the virion, to one of the smaller proteins that hasn't been released yet, through its light chain. It uses a nuclear porin to bind to its heavy chain of that kinesin, conventional kinesin. And that heavy chain binding is activating the kinesin. It's increasing its ATP turnover. And when a microtubule happens to be near the nuclear pore, which happens, the kinesin starts to recognize the pore and starts to walk. Now it walks together with the virion, or tries to remove the virion, but the virion is tethered to the nuclear pore by another protein, by the hexon protein, which binds to another nuclear porin. And so that creates a sort of, if you will, a tug of war on the virion, and it leads eventually to the disruption of the particle and liberates the DNA. Now the DNA that's yet another thing. It's interesting actually. Not all the DNA is finding the way into the nucleus. Some DNA gets misdelivered. It ends up in the cytosol. And that's an interesting feature that happens and may contribute to the inflammatory nature of these adenoviruses that now put a danger signal into the cytoplasm.
Sure.
And especially if you're an immune cell, of course, then you are equipped with translating this danger signal into an antiviral response and maybe an inflammation.
So the DNA will be sensed in the cytoplasm for sure. Yeah. But that's a mistake as far as you can tell. There's no point in putting viral DNA in the cytosol, right?
Well, if you will, yes, it's an inaccuracy, if you will, maybe from the virion's point of view. And we missed this phenotype. This is very interesting, actually. We missed this phenotype by our initial assays, which were done based on in situ hybridization of the incoming viral DNA. In this protocol, we had to use heavy chemical conditions to dissociate the double-stranded DNA into single strands to make them available for the probe, the fluorescent probe, to see them. But in that procedure, it turned out that we have removed essentially all the cytoplasmic DNA due to the chemical extraction conditions, so pH 1 also. So we concluded that we saw viral DNA in the nucleus, and we were happy with this, right? So that's what you expect. And but we missed completely that the virion is also misdelivering things. So this misdelivery observation only came to notion when we were collaborating with chemists who gave us tools to use click chemistry to label the incoming virion DNA in a non-invasive manner, in a bioorthogonal reaction that you could track without heavy disturbance of the cell the incoming DNA. And there we saw that the DNA is indeed misdelivered, and the misdelivery happens in a largely, in a huge variability, with huge variability from cell to cells. So there are cells that have almost no misdelivery, and there's others that have a lot of misdelivery. And that's a puzzle. And it relates to one of our questions that we have in trying to better understand the cell-to-cell variability of how these cells react to virus infections and how viruses can deal with this.
So is it less misdelivery in, say, a respiratory tract cell compared to a cancer cell line?
That we don't know. We have not systematically analyzed these primary cells. So you would wish to do a maybe an organoid type of experiment and then look at things. But this is challenging, and we haven't really proceeded to that level of detail.
I just wonder if it's more accurate in the physiologically relevant cell as opposed to a cell line. It would be interesting to look at. But I mean, there are cytosolic sensors for DNA, and they always troubled me. Their existence troubled me because, in my view, DNA viruses perfectly transport, except for some that were replicated in the cytosol, they perfectly transport the DNA to the nucleus. But now we see it's not perfect, and that's probably why those sensors exist. And they probably tell us that in the real cells where these viruses infect, there is leakage, there's mistakes made. Yeah.
Right.
Do you know of other viruses that misdirect the DNA into the cytosol, for example?
Yeah, so a student in the lab has looked into a little bit of herpes virus. There's not much misdelivery, but there's maybe a little bit there also.
Yeah. Okay.
Systematically, we haven't looked into all these other DNA viruses.
So I want to point out a review article you wrote called Principles of Virus Encoding, Cues and the Snooker Ball. Do you play pool?
Sorry?
Do you play pool?
Not on a regular basis, but of course I like that, yes, of course.
Yeah, so what you've said today is the cues, including the ones around the nucleus.
So exactly, the cues are those things that happen directly with an impact on the virion. And the virion would be the ball, right? That is in this game. And upon the cue, the ball is doing something, is maybe finding the way to the hole. And if it's successful, then the next step can happen.
It's nice. I like that. So there's one other paper you've written which I'd like to discuss because I'm a big fan of plaque assays, as people may know, right? It's called Plaque 2.0, a high throughput analysis framework to score virus cell transmission and clonal cell expansion. Plaque 2.0. Can I make a wall of Plaque 2.0? Probably not. So what is Plaque 2.0?
It's an assay that uses not the classical kind of analysis of a plaque, namely on a petri dish, but it's a microscopic assay of a plaque assay, and it picks up a phenotype of the plaque that is before the cells that have been infected have lysed in that area. So it gives you more information about how the plaquing occurs, and also it gives you information about what kind of plaques there are. There's different kinds of plaques. There's not just small and big plaques. There's also comet-like plaques and round plaques, because one can do this assay without agarose. So agarose is used classically in the plaque assay to restrict the convective flow of fluids in that area. You can do this in a live assay without this agarose, and you will get information about the way virions travel from cell to cell based on the shape of the plaque.
So it's really a fluorescent focus assay, which I remember. Do you remember Hamish Young? Do you know the name? It's an old adenovirus. He's retired many years, but he was at Columbia, and he would always say, I don't want to wait two weeks for the plaques to form. So after a few days, you can stain with an antibody and look in the microscope and plant foci. So this is basically a mechanized way to do that, right?
Yes, it's a high throughput way. It is unbiased because you sample the entire well that you decided. You can do it in 96 well formats or even in smaller formats. You sample the entire information there in a time-resolved manner.
And you don't need the cells to die. That's the key, right?
Not necessarily.
Just make proteins.
Exactly.
Yeah.
And the assay is powerful because it allows you now to sample the entire life cycle of the virion in a fluorescent assay. And you can do new things with this.
How would you do the whole life cycle? By labeling proteins that are made at different times? Is that what you think?
Yes, you can make transgenic viruses, obviously, or you can decide to make an endpoint assay where you have let's say, plaque formed in many instances, and you can just decide then to score the shape and the size and the numbers of these plaques depending on your interference conditions. And you can see, that the first time we did this assay was with a student in 2012. It was a J-Virol paper on that one, where we noticed that besides the plaquing phenotypes that one sees in these dishes, there are numerous cells that were infected but were not plaquing, and this was based on a GFP reporter virus that was replicating and making the full cycle.
Right.
So that was striking because it told us, look, only about 10 to 20% of the cells were making the plaque. The rest of these cells at this time point were infected, or some of them were infected, but not making the plaques. So what is happening there? Right, so it's an incredibly interesting biological question now that you can attack from the host side, right, like Silke would do maybe, or from the virion side also.
So are other labs using this approach to look at different viruses, or is it just you so far?
Yes, so we did get requests for the protocol, for essentially for the bioinformatics from quite a few different labs. So I don't know exactly the numbers. So Arthur would have to tell me, the student who did that. He's now a postdoc at UCL, but I think he's keeping track on that. And so we have transplanted this assay from the University of Zürich down to the EPFL, that's the Swiss Federal Technical University in Lausanne, where they are interested in drug screening as well, and they are now using this assay as well for scoring chemicals that would interfere with the entire life cycle of virus infections.
I have to point out that UCL is University College London, right?
Correct.
And that's Imperial College London is a different place, right? It's an inside joke. You have to listen to TWiV to learn that. So you have, this is done in cells just like a regular plaque assay, except you use transgenic viruses, and then you have a machine that looks at the plate. It's a microscope attached to a scanner, basically, right? And it automates, you can automate the scanning of many, many plates. Yes. So in the end, you do have plates. So you have yes. 384 well plates instead of yes. 6 well, right?
Yes.
So, in my office, I have a wall of 6 well plates. So you could still do that with this assay, right?
We could. It would just not be nearly as impressive as your wall because we have much fewer of these plates.
Yeah, because you have 384 wells, yeah. See, that's the only thing I worry about, that you could make a wall of polio with it. But I really think it's an amazing approach. And this is really a reflective of how microscopy and automation has changed since you were in Ari's lab, right?
Oh yeah, that's changed a lot.
Huge.
The first experiment, I remember when we did that, the first live experiment in my lab was with a stopwatch. And with a manually controlled slider of the microscope where we switched between red and green colors with a stopwatch. And that gave us the first movies, and it told us, it was interesting nonetheless, it was informative because we found that viruses were behaving in terms of their movement very heterogeneously in cells. Sometimes they were moving rapidly, very fast on microtubules, and sometimes they were stalling. And this kind of information we could read out, but we had no idea what it means.
Well, if you look in the textbook of Principles of Virology, we still have one of your original figures, which we got from Ari, which is the adenoparticle docked on a nuclear pore. It will always remain there because that's obviously right. I want to say that, this is interesting, this stopwatch, but before there were PCR machines, we would have water baths at different temperatures. You put a tube in one, a tube in the next. You do it by hand because we didn't have these automatic cyclers.
Absolutely.
And of course, I used to pour these huge sequencing gels, which we don't have to do anymore. So technology is great to push science forward. And, I've had a relatively short period of research. There are other people who have done it longer. But when you look from 1970s to the present, it's just remarkable. The only thing I had was recombinant DNA technology. That was brand new. And everything since then has just made it so much easier to do experiments. It's just amazing. That's why science is wonderful. I want to ask all of you a few quick questions before we wrap up. Silke, if you had not been a scientist, what would you have done?
Oh dear. Maybe a vet?
A veterinarian taking care of animals?
Yes, I think that could
Small
be.
animals or big animals?
Small.
Kitties, like cats, yeah, okay.
Yeah, maybe that.
All right. Cornel, what would you have done?
I would be a geologist.
A geologist?
Yes.
Interesting.
If I retire, I go study geology.
Okay. Rocks. Urs, what would you have done?
I can't think of anything else than being a scientist.
Okay.
Sorry.
That's fine.
It's
Actually,
it's fascinating and it's fulfilling and keeps you going and keeps you in touch with new things all the time.
Silke, what do you think is your most important contribution to science so far?
Ooh. I would hope that our studies on host factors that could potentially become a drug target, that will eventually be of some benefit, even if it's just to see that this is not a strategy that works. But I think this direction to explore that, I think, would be helpful.
About you, Cornel?
I hope it's my students that go out and become scientists. And the second is probably my contributions to live cell microscopy for replication, and also earlier things like cloning large virus genomes as bacterial artificial chromosomes to do genetics in bacteria and create viruses in after transfection cell culture.
Urs, what about you?
Well, it's probably something in virus entry. And I agree with Cornel. I think it's fascinating to see the students and postdocs going away and continuing in science and doing other things there.
Okay, let me ask you the next one, so these can have some time to think about it. Can you tell us some inspiring book you've read lately?
Well, a couple of books maybe come to mind. So Thomas Friedman, The World is Flat, I read. It's an old book, actually, and the newer one, a couple of years old, is Sorry I'm Late, or Sorry for Being Late. And I think this kind of book, opening up new ways, for me at least, into other worlds, into the world of rapidly moving on in different areas besides the biological sciences is hugely interesting. And so I was deeply impressed by these books. And I found them very exciting.
Cornel?
Well, I stopped reading philosophy books. My way now into other worlds is, at the moment, I mainly read books on hunting boars and
There are books on hunting boars?
Yes.
You have a lot of boars in Switzerland?
Yeah, quite a few.
And do you eat them after you hunt them? Or do you put them in the lab and look for...
Both, actually both. We have a very important project now at the Institute where we look at hepatitis E viruses in boars,
Right.
some of which I kill. And we found that about 30%, you correct me if I'm wrong, 30% of these animals are Hep E seropositive.
Right.
And a large number of the animals also are virus positive. And we started doing a complete virome analysis of these boars and also some deer.
Mm-hmm. So you think that that's one way that people may get hepatitis E infection?
Oh yes, definitely yes. It's from undercooked food, meat products, liver products. And so that's my contribution to this study.
Do you cook the boar really well?
No, I test them before I eat them.
Oh, you check? You have a PCR machine at home? So Urs, last night we had deer.
Yep.
And it was very red, so I was worried about it.
Yep.
Did you ever look for Hep E in venison?
Hep E, yes. We did look at some, but they're all negative. So good for you.
I almost wanted to send it back to the kitchen, but that's kind of insulting to the chef, right? Okay, how about you? A good book?
Going to be very boring after Cornel's choice. I have a little son, so I have very little time to read for myself. I read mainly children's books about Father Christmas at the moment.
Father Christmas?
Yes.
Do you ever read science children's books?
We got one about the I think it's called The Fascinating World of Microbes, is still a little bit tricky, but he seems interested by these little things in that book, but that might come.
A colleague of mine in Mexico, Susana Lopez, she's a virologist, she just worked with a team of virologists and a graphic artist to make a children's book.
Saw that, yeah.
Paul has measles. It's really good at explaining viruses and measles and vaccines in particular to kids. It's free. You could download it. So you should check it out. All right, one more question. DNA or RNA?
RNA.
DNA.
In terms of what?
Oh, don't ask. Don't ask. Just, it's a quick response.
RNA.
Oh, I'm surprised. Very good. Very good. All right, thank you. That's TWiV 523. You can find TWiV on any podcast player that you use to listen on your phone or tablet. There are many. Just search for TWiV and subscribe. We'd like everyone to subscribe to the podcast, even if you don't want to listen. But of course, you do want to listen, because when a new episode is released, you get it automatically. It's downloaded, and we see that number, and it turns into numbers of downloads. That's happened before. That's me. So that's not the first time. Subscribe so that, thank you for subscribing. You get every episode as it's released. And we love getting questions and comments. We didn't read any on this episode, but we love to discuss them. TWiV@microbe.tv. If you really like what we do, contribute financially. You can go to microbe.tv/contribute. We have a number of ways you can do that, including Patreon. My guests today have been from the University of Zürich. Silke Stertz, thank you so much.
Thank you for having me.
Cornel Fraeful, thank you so much for coming.
Thank you very much.
And those boars, man, that's really something. And Urs Greber, thank you so much for joining.
It's a great pleasure, thank you.
And this is just an example of the cool science that goes on here. There's much more. And you should have me back another time to do more. I'd be happy. We do other podcasts besides virology, microbiology, parasitology, and so forth. I'm Vincent Racaniello. You can find me at virology.ws. I want to thank Anita Meyer for arranging this visit. She first emailed me a year ago, and it's a year later I've come. It was really hard. I had all kinds of requests and problems, and she just dealt with it perfectly, so thank you so much for having me. Serena for picking me up at the airport. Thanks so much. And the LSSS Life Science Seminar Series, which supported my visit. I want to thank ASM for their support of TWiV and Ronald Jenkins for the music. You've been listening to This Week in Virology. Thanks for joining us. We'll be back next week. Another TWiV is viral.